Hello dear learners, today we will see the quality control test for parenteral preparations. As in the all previous video, we have already seen the details of parenteral preparations. Now this is a very important how we can evaluate the parenteral preparations. So we will see the QC test for parenteral preparations. Now the type of quality control test. So there are generally two types of quality control test. One is in-process quality control test and finished product quality control test. So in process, it includes like conductivity, pH measurement, then volume filled, temperature for heat sterilized product, and environmental control test, or we can say the osmolarity. Now in the finished product QC test, these are the examples of tests that we have to see in detail and we have to study in detail. Now we will see first the in-process quality control test. Now we will see first is the conductivity measurement. So obviously conductivity measurement is done by conductometer and it measures the conductivity of vehicle that we have to use to prepare parenteral preparation, or we can say the sterile preparation. Now the next is pH measurement. So different types of methods used in measurement of pH. Like we can use pH meter, digital pH meter, or we can dip a piece of pH paper into the sample. Now the next test is the temperature for heat sterilization. So this is the very important to maintain the constant temperature during heat sterilization of product. So the temperature changes may cause some undesirable changes like change in potency, then change in isotonicity. So we have to maintain the temperature during sterilization for heat sterilization. It is very important. So the temperature can be determined by normal thermometer. Now the next is environment control test. So here we should maintain the environment. We should control the environment. So personnel should be permitted into aseptic area only after following the prescribed procedure. For example, surface disinfection personnel should be in a good health. Air control should be there, like HEPA filter. Then it is 99.97% efficient, removes the particles of 0.3 micrometer size and larger, and velocity should be 100 plus 20 feet per minute. Now the next is volume filled. This is a very important test. Means how we can measure the volume filled. So for that, what we can do, that injection container is filled with a volume in slight excess of the label value. For example, if the label value is 0.5 ml, the mobile liquid is 0.1 ml, so viscous liquid should be 0.12 ml. As like that, we have to maintain the mobile liquid as well as viscous liquid with this label size. Now we will see the very important test is a finished product quality control test. The first one is Content Uniformity Test, then leaker test, or we can say the leakage test, then Pyrogen test, Sterility test, as well as Particulate test. So the first one is Content Uniformity Test. So in Content Uniformity Test, how we can perform it? So firstly we have to take these 30 sterile units are selected. Means we have to take 30 containers or products from each batch. The weight of 10 individual sterile units, 10, we have to weigh the 10 containers is noted and the content is removed from them. An empty individual sterile unit is weighed accurately again. We have to first weigh the filled container and then empty container. So then net weight is calculated by subtracting the empty sterile unit weight from the gross weight, means the filled container. So the dose uniformity is met if the amount of active ingredient is within the range of 85 to 115% of the label claim. Now how we can confirm, how we can say that the content uniformity test is passed or failed. So if the relative standard deviation is equal to or less than 6% and if one unit is outside the range of this limit and none of this sterile unit is outside the range of 75 to 125%. So if the relative standard deviation of the resultant is greater than 6, or if we can say this both, the condition prevail, then an additional 20 sterile units should be tested. Means what? If this both conditions are not met, then we have to test another 20 sterile units. And what we will get after that? So the sterile units meet the requirements if calculated relative standard deviation is not more than 7.8%. So this is how we can perform the content uniformity test. Now we will see the leakage test, or we can say the another name is a leaker test. So this is the instrument. It's a digital instrument to measure it, to apply the vacuum. So what we have to do over here, how we can perform. Firstly, why there is a need to perform this test. So we can say that it is generally applied to ampules because we have to detect incompletely sealed ampules so that they can be discarded in order to maintain the sterile conditions of the medicines. The tip seals are more likely to be incompletely closed than pool seals. So open capillaries, or we can say cracks at the point of seal, results in the leakers. That's why we have to perform the leaker test. So here what we have to do, how we can perform it. So this test is performed by immersing the ampules in a dye solution. We have to check in the ampules leakage is there or not. So what we have to do, we have to immerse the ampules in a dye solution such as 1% methylene blue and applying at least 25 inches of vacuum for a minimum 15 minutes. Detection of leaker is prominent when ampules are immersed in a bath of dye during autoclaving. We have to do autoclave it, as this has advantage of accomplishing both leaker detection as well as sterilization. It is a high frequency spark test. Detect presence of pinholes in ampules. So we get the color solution from the transparent. Means if we will get the blue color in our container, then we can say that yes, there is a leakage. That's why this color of methylene blue came inside in the ampule. Now we will see the next is a Pyrogen test. So this test involves the measurement of rise in a body temperature of rabbits. Here we have to use rabbits following the IV injection of a sterile solution into ear vein of rabbit. Here, whatever dose we are using, it will not exceeding 10 ml per kg injected intravenously within the period of not more than 10 minutes. And what are the animals? We have to use healthy adult rabbits of either sex, preferably of the same variety. Recording of the temperature is done by clinical thermometer or thermistor. Now we will see how we can perform the Pyrogen test. Now we will see how we can perform the Pyrogen test. So there are two tests. Firstly, we have to do preliminary test. If the rabbits are eligible for preliminary test, and if the preliminary test is passed, then and then only we will go ahead for a main test. Now first what we have to do, if animals are used for the first time in a Pyrogen test or have not been used during 2 previous weeks, we condition them 1 to 3 days before testing the substance by injecting IV 10 ml per kg Pyrogen free saline solution, which is warm to about 38.5 degrees centigrade. Then record the temperature of the animal beginning at least 90 minutes before injection and continuing for 3 hours after injection. If any animal showing a temperature variation of 0.6 degree or more must not be used in a main test. So if we will observe any animal which will have temperature variation of 0.6 degree, then we will not enter that particular animal into the main test. So this is the main test. We can see over here the group of rabbits. So this test is carried out in a group of 3 rabbits. How we can prepare a solution? So for preparation of the sample, we have to dissolve the substance in or dilute with pyrogen-free saline solution. Then warm the liquid to approximately 38.5 degrees centigrade before injection. After that, what we have to do, inject the solution under examination slowly into the marginal veins of ear of each rabbit over a period of not exceeding 4 minutes. After that, we have to record the temperature of each animal at half an hour intervals for 3 hours after injection. And the difference between the initial temperature and the maximum from this 3 hour, whatever temperature we will get, the maximum that we have to take, which is the highest temperature recorded for a rabbit, is taken to be used for its response that we have to take. So we can perform the pyrogen test like this. Now we will see the next test is a bacterial endotoxin test. It is a very important test. Bacterial endotoxin test, what is it? So it will measure the concentration of bacterial endotoxin that may be present in the sample using a lysate. We have to use a lysate. The addition of a solution containing endotoxins to a solution of a lysate, which will produce turbidity or precipitation, or we can say gelation of the mixture. That's why we have to use lysate. These are the methods to perform the bacterial endotoxin test. First will the method A, it's a gel clot limit test method. Then method B is a semi-quantitative gel clot method. Method C is a kinetic turbidimetric method. Then method D means a kinetic chromogenic method, and method E means endpoint chromogenic method. Now we will see in detail. So what is gel clot limit test method? Here what we have to do, we have to prepare a sample solution at any dilution, at or below maximum valid dilution. Then we have to use the water bacterial endotoxin as a positive as well as negative. We have to take 2 positive control and 1 negative control. One of the positive control consists of a controlled standard endotoxin, and other consists of a test solution spiked with controlled standard endotoxin. After that, we have to see what happened, how we can measure it. So the product under examination complies with the bacterial endotoxin test if the positive control is positive and negative control as well as test solution are negative. And if we will get the result like the test is not valid if the positive control is negative and negative control is positive, means whatever solution we have taken, that should be same after testing. Now the next is kinetic turbidimetric method. So here what we have to do, a photometric assay measuring the increase in a turbidity, or we can say turbidimetry, caused by the reaction of the endotoxin with a lysate. There will be a reaction between these two. That's why we can produce or we can get a turbidity. Now in kinetic chromogenic method, what we have to do, we have to add some material or substance, we can say, which can produce a color because it is a chromogenic material. So photometric assay measuring a color developed by the chromophore released from a chromogenic substance by the reaction of endotoxin as well as lysate. So we can find from turbidity as well as the production of color there is a presence of endotoxin or not. Now we will see the sterility test. So sterility test is defined as a microbiological test that is applied to sterile product to show that our product manufactured and processed under specification guided by CGMP. Now see what we have to use for sterility test and what are the method. So media to be used in a sterility test, there are 2 medium. FTM, it's a fluid thioglycolate medium, and SCDM means soybean casein digest medium. What are the methods? There are 2 methods, method A as well as method B. So method A is a membrane filtration method and method B is a direct inoculation method. Now we will see this methods in detail. So first is a membrane filtration method. So what is it? So here we have to do filtration only, but we have to perform it by membrane filtration. Means a membrane has a nominal pore size not greater than 0.45 micron and diameter of approximately 50 mm we have to take, and this method basically involves the filtration of a sample through the membrane filters. So the filtration is assessed under vacuum. We have to perform filtration under vacuum, and after completion of filtration, the membrane is cut into two hulls, and one hull is placed in two test tubes containing FTM, fluid thioglycolate medium, and soybean casein digest medium. After that, we have to incubate the media for not less than 14 days. Now, the next is direct inoculation method. So, this test involves a direct inoculation of required volume of a sample in two test tubes containing a culture medium of the same, this FTM and SCDM. Volume of the preparation under examination is not more than 10% of the volume of the medium, and incubate the inoculated media for not less than 14 days. After that, we have to check it. Now, what are the minimum number of items to be tested? So here for parenteral it is given the number of items in the batch and minimum number of items to be tested for each medium. So here the number of items, it's not more than 100 container. Next is how we can interpret the results. So interpretation of results. So how we can interpret? If the material being tested renders the medium turbid so that the presence or absence of a microbial growth cannot be easily determined by visual inspection, means after 14 days. So 14 days after the beginning of incubation, then transfer the portion less than 1 ml of the medium to fresh vessels of the same medium, and then incubate original and the transfer vessels not more than 4 days. If no evidence of microbial growth is found, then it complies for test for sterility. And if there is evidence of a microbial growth, then it not complies for the test for sterility. This is how we can measure or we can interpret the results. Now we will see the last is a particulate test. So how we can perform the particulate test? There are two methods to find the or count the particles. First is method A and method B. So method A means the light obstruction particle count test, and method B means microscopic particle count test. The first is light obstruction particle count test. So here we have to use a suitable apparatus based on a principle of a light blockage, which allows an automatic detection of a size of particle and the number of particle according to size. So here we have to measure two things, the number of particle and size of particle. So we have to pass the light like this. Here we have to detect our particle from passing the light. Here there is a reflector, the light source as well as the light trap. Now the next is the limits. So this is the limit for particle size in a micrometer, for sample the LVP, and for SVP. These are the maximum number of particles should be there. Now the next is the microscopic particle count test. So in this what we have to do, wet the inside of a filter holder fitted with the membrane filter with several millimeter of a particulate free water. Then transfer the total volume of a solution pool or of a single unit to the filtration funnel and apply the vacuum. After that, we have to place the filter in a petri dish and allow the filter to dry air, or we can say air dry. After the filter has been dried, place the petri dish on the stage of microscope and scan the entire membrane filter under the reflected light. This test we have to perform into the microscope and the first one from the direct light. This is the difference. And count the number of particles. So this is the limits for that particular test. So here The in-process and finished product quality control test is completed. Now in the next video we will see the ophthalmic preparation. Thank you dear learners for watching this video.